How many animals do you see here? In truth, we see only a fraction of the animal world that actually exists. Larger animals are the most noticeable. Smaller animals, like many insects, often go unnoticed. But insects in particular are by far the most species-rich group of all living creatures. They make up about 70% of all animal species worldwide. Insects are not just a nuisance or harmful. On the contrary, insects have irreplaceable valuable function in ecosystems. These include bees and butterflies, as well as beetles and ants. They pollinate plants, control pests by eating other insects, and themselves serve as a food source for other animals such as birds. Insects are therefore crucial to the functioning of ecosystems, including cultural landscapes shaped by humans, such as agriculture. However, insects are currently under severe threat. A recently published study shows that the biomass of flying insects in protected areas in Germany has collapsed by over 75% in a period of 27 years. To protect biodiversity, targeted measures must be implemented. These measures are based on information obtained through monitoring, which is the surveillance of changes. In order to determine insects' biodiversity in open areas, insects are either identified by experts directly in the field by hearing or seeing them, or insects are captured and later on identified in the laboratory. Frequently used passive methods are, for example, malaise traps. Insects fly against the net, which is invisible to them, and try to cross this barrier. In this process, the insects are flying up and down. As it flies up, the insect is directed into a collection vessel. The vessel is filled with ethanol, which kills and preserves the insects. An alternative is offered by eDNA methods. eDNA stands for environmental DNA. All animals, even those that are tiny and live in hiding, leave DNA traces in their environment. This happens in the form of, for example, skin remains, scat, or saliva from feeding traces. To determine which animals are present, samples need to be collected from the ecosystem of interest. For example, flowers, leaves, soil, or water. Each sample contains genetic information about different species. Like humans, each species has a fingerprint in the form of a specific genetic sequence. The small amounts of eDNA in the samples can be specifically detected using the method of metabarcoding. In the process, the sequences are amplified using a polymerase chain reaction and then sequenced. The resulting eDNA sequences are aligned with a reference database for matches, thus identifying the different species. Classic monitoring methods often interfere with the ecosystem and kill the animals. However, the eDNA method makes it possible to detect the animals without having to see, hear, or capture them. The simplicity of the eDNA method is an attractive addition to classical monitoring methods and can therefore also be used as a potential citizen science tool. eDNA methods help to better understand ecosystems and contribute to their protection.